Synaptic transmission. The synapse is a structure that allows chemical communication in neurons. It consists of the presynaptic neuron, the postsynaptic neuron, and the synaptic cleft that separates the two neurons by a small extracellular space. The structures involved in synaptic transmission are the synaptic vesicles containing neurotransmitters, the calcium ions, calcium channels, ions, and ion channels. To initiate synaptic transmission, an action potential arrives at the presynaptic axon terminal, triggering the opening of calcium channels on the presynaptic membrane. Calcium ions flood into the presynaptic neuron and bind to nearby synaptic vesicles. Synaptic vesicles that have been activated by the binding of calcium ions fuse with the presynaptic membrane and empty their contents into the synaptic cleft. The neurotransmitters cross the synaptic cleft and bind to receptor proteins on the postsynaptic membrane. If the neurotransmitter binds to sodium ion channels, the membrane potential of the postsynaptic neuron is brought closer to the threshold for generating an action potential. This is the activation or stimulation of a postsynaptic neuron. If the neurotransmitter binds to potassium ion channels, the membrane potential of the postsynaptic neuron is brought further away from the threshold for generating an action potential. This is the inhibition of a postsynaptic neuron. After the channels on the postsynaptic membranes close and the neurotransmitters are released back into the synaptic cleft, they are quickly destroyed by enzymes or taken back into the presynaptic terminal by endocytosis in a process called reuptake. The destruction or reuptake of neurotransmitters prevents their effects on the postsynaptic neuron from being too long-lasting.